Welcome to our channel Mootechie. Today we're going to explore the fascinating kingdom of fungi. Let's begin with the general characteristics of fungi. Fungi are eukaryotic, non-photosynthetic organisms that absorb nutrients via saprotrophic or parasitic modes. This means they cannot make their own food like plants. Instead, they get nutrients by breaking down dead matter or living off other organisms. Their cell walls contain chitin, not cellulose, and their primary storage carbohydrate is glycogen. Chitin gives their cells strength and rigidity, similar to how cellulose does for plants, while glycogen is the same energy storage molecule found in animals. The fungal body consists of filamentous hyphae forming a mycelium, though unicellular yeasts also exist. Think of the mycelium as a vast, branching network hidden in the soil or food, while yeasts are single, free-living cells. They reproduce by spores, which can be asexual, produced by mitosis, or sexual, produced by meiosis. Spores are like tiny, durable seeds that allow fungi to spread and colonize new areas. Fungi are mostly terrestrial and act as critical decomposers or mutualists in ecosystems. Without them, ecosystems would collapse under piles of dead leaves and wood, and many plants would struggle to get nutrients. Now, let's look at the basis of fungal classification. Classification is based on morphology. This distinguishes between unicellular yeasts, filamentous molds, and dimorphic fungi which change form based on temperature. For example, some human pathogens are yeast-like in the body, but mold-like in the environment. It is also based on reproduction, looking at asexual spores like conidia versus sexual spores. Key groups are defined by their sexual stage, producing unique spores like zygospores, ascospores, or basidiospores. Hyphal features are also important, such as the presence of cross walls, making hyphae septate, or having continuous cytoplasm called koinocytic. Unique structures like clamp connections are also key identifiers. Finally, modern classification relies heavily on molecular data like rRNA and phylogenomics, as well as ecological roles or host range. DNA analysis has revolutionized how we understand the relationships between different fungal groups. Here, we examine the cell structure of fungi. First, the cell wall and membrane. The rigid wall is composed of chitin for strength, and the cell membrane is rich in ergosterol, which is a target for antifungal medications. Next, the hyphal structure. Hyphae can be septate, meaning divided by cross walls, or koinocytic, with continuous cytoplasm. Growth occurs exclusively at the hyphal tip, which is why fungi spread outwards in a radial pattern. Fungi contain key organelles. Typical eukaryotic ones like nuclei, mitochondria, vacuoles for storage and turgor pressure, and peroxisomes. They also have specialized structures. These include septal pores for cytoplasm flow between cells, haustoria for nutrient absorption from hosts, and reproductive sporangia that produce spores. This slide also includes another diagram labeled input showing structures like the mouth, mastax, pseudochole, stomach, intestine, and anus. This again depicts a rotifer's digestive system, emphasizing how it processes food from ingestion to excretion. We'll now start covering the major classes of fungi, beginning with part 1. First, Chytridiomycota, known as chytrids. These are mostly aquatic fungi that retain flagella, a rare feature among fungi. They produce flagellated zoospores for movement in water. An example is Batrachochytrium, a notorious amphibian pathogen causing chytridiomycosis. Next is Zygomycota. These are fast-growing molds with koinocytic or non-septate hyphae. They form resistant, thick-walled zygospores during sexual reproduction. A common example is Rhizopus, the black bread mold you might find on spoiled food. Then we have Ascomycota, the sac fungi. This is the largest group, including yeasts, truffles, and molds. They produce their sexual spores, called ascospores, inside a sac-like structure called an ascus. Examples are Saccharomyces, the brewer's yeast, and Penicillium, the mold that gives us penicillin. Finally, Glomeromycota. These are ecologically vital obligate symbionts, meaning they must live in partnership with another organism. They form arbuscular mycorrhizae with plant roots, deeply penetrating the root cells to exchange nutrients. An example is Rhizophagus. Let's continue with part 2 of the major classes. First, Basidiomycota, the club fungi. This group includes mushrooms, rusts, and smuts. They are mostly sexual and produce their sexual spores, called basidiospores, externally on a club-shaped structure called a basidium. Examples are Agaricus, the button mushroom, and Puccinia, a type of rust that affects plants. Next, we have Microsporidia. These are unicellular obligate intracellular parasites of animals. They have highly reduced organelles, lacking normal mitochondria, and produce spores to infect new hosts. An example is enterocytosone, which can be a human pathogen. We also have deuteromycetes, known as fungi imperfecti. This is an artificial group for fungi where we have only observed an asexual stage. They reproduce only via asexual conidia or conidiospores. Many Penicillium and Aspergillus species are classified here until their sexual stage is discovered. Finally, we have the special growth form yeasts. This is not a taxonomic class, but a unicellular growth form found primarily within Ascomycota and Basidiomycota. Examples are Candida albicans, a common cause of yeast infections, and Cryptococcus, which can cause serious lung and brain infections. Now, let's discuss the immense importance of fungi. First, as decomposers. They recycle carbon and nitrogen and sustain soil health by breaking down organic matter like dead plants and trees. In food and fermentation, they are essential. We use them to make bread, beer, wine, and cheese, and to produce organic acids like citric acid. In medicine and biotechnology, fungi give us life-saving antibiotics like penicillin, immunosuppressants like cyclosporin for organ transplants, and cholesterol-lowering statins. In agriculture and ecology, mycorrhizan fungi enhance plant nutrient uptake, and fungi like trichoderma are used as biocontrol agents against plant diseases. However, fungi also pose risks. They cause devastating plant diseases like rusts, mycosis or infections in humans, and produce harmful mycotoxins such as aflatoxin, which can contaminate food. Thank you. We've explored the basis of classification and the diverse major groups of the fungal kingdom. Here's a challenge for you. Can you identify a fungus's class just by looking at its spore type and hyphae? Try observing a mushroom or mold and see what clues you can find. We hope this overview has helped illuminate the incredible world of fungi. Remember, these organisms are all around us, playing silent but crucial roles in our lives and the health of our planet. Keep observing and stay curious.